I'd like to introduce Dr. Julius Jacobsen, who is part of the Monarch Initiative, but also senior software developer at the Centre for Translational Bioinformatics at Queen Mary University of London. He's worked at the Sanger Institute, at the European Bioinformatics Institute, and he's a member of the Phenogenomics group at the William Harvey Research Institute at Queen Mary. But for these purposes, particularly, he's the lead developer of the Exomizer toolkit, and the lead for the kind of work on phenopackets as well. So I think that's what we're going to focus on. So thank you very much, Jules.
Thank you very much for the introduction. All right, hello. Thanks for having me here. So I'd just like to say we've been working with Peter and Damien over there for about over a decade now. So this is all very much a collaborative experience that we've been going through, and all this work is not just my work; it's very much a lot of other people's work as well. So this is a huge amount of people coming together to create all of this. So I'm going to be talking basically in two halves about two different things, but they're quite allied to each other. So we've got the HPO in action. So we've got phenopackets, which is how you apply the HPO in a kind of more as a packaging, just to sort of keep your phenotypes together in a structured format so that you can share it with people. And then with the Exomizer is more about actual diagnostics, so how you would use that data and to actually come up with a diagnosis. So the first thing you need to do in these days is you have to say, ChatGPT, tell me about this thing because I have no idea what it is. So you might have heard the term phenopacket, and this is actually genuine stuff. I tried this yesterday and it came out quite well. So I thought I'd keep it in here because it's like, how do I describe this? So it's a standardized digital container for sharing detailed phenotypic data about individuals, typically for biomedical research and clinical practice. So we're looking really for exchanging health information about clinical symptoms, history, medical history, and specifically with that genetic information tied in with all of this. Yeah, as I said, it's quite in there for sort of genetics, rare disease research, and personalized medicine. So what are the use cases for phenopackets? I'll go into a little bit about the structure of the phenopacket in a while, but we'll sort of go from a high-level view about why are we using these things. So there are a lot of people who want to actually describe a phenotype of a patient in lots of different contexts. And really, before this, there was no real way of doing it. You could have an EHR system where you'd be able to put the data in here. But again, the EHR is obviously tied to the EHR. It's not outside of this thing. There are also the data warehouses that you can use to keep this data in there. But again, they're stuck in a data warehouse. So the phenopacket is really meant to be sort of external of these things in a free format that people can use to exchange as easily as they need to. So the people who want to use this would be sort of physicians to actually describe the patient, patients who maybe want to describe themselves, or the families of the patients, if they specifically for rare diseases where you have a completely undiagnosed condition and you want to go out there to say who else is looking a little bit like this. As soon as you have a phenotype that you can say the similarity to other patients, then maybe you have a similar genotype there as well, so you can start to solve cases this way. Obviously, when you come to reporting new findings, the researchers and authors would want to be actually pulling the data they have together to have it in a structured format that you can then disseminate to other people. So like you would have a DOI, for example, that would be a piece of structured data to describe how you find a document. In this case, you'd have a linked document to this, which would be the actual phenopacket to describe the patient within the actual references themselves. So laboratories obviously would find this sort of information useful because they need to have information about the patient's phenotype in order to make a diagnosis if they have any kind of genotype information to find a causative variant. This isn't always easy to get out of an EHR. So the idea we have here is with the phenopacket, the real key thing here is standards. So if you have a standardized way of providing your information and packaging up, then you have a standardized tooling that you can apply to it. These become portable and data becomes a lot easier to share and a lot freer and a lot more easy to actually use. So as Peter was saying beforehand, so there are a lot of different use cases for these things. So he was using it beforehand to show patient stratification to actually find new disease gene associations. You can use it for identifying cohorts of patients to say which ones are more similar to others, and then lump those together, and then maybe use those for whatever needs you might have. And then there's for diagnostics and decision support. So I mentioned EHRs, and you can see on there there's this OMOP and FHIR. So these are two fairly large players in the EHR and clinical space. So HL7 Fire, I think, is the lead on from the V2 messaging that they used beforehand. This tends to be very much part of the EHR themselves. There is a huge amount of tooling behind it, and it's a very large and very well used, but also very heavyweight and complicated piece of software to have in there. And there's a lot of tooling required to get these things to work. It is a standard, and it's extremely well supported, and it's very good, but it lives really within the EHR is the kind of the space where that works in. Then sort of allied to that, you've got Odyssey, which is the observational health data sciences informatics. They've got OMOP, which is their common data model they have, which is a set of database schemas. So again, this has to live as a set of database tables in the database. You can query this. The difference between these two here is that Fire is really working about individual pieces of information about a patient, whereas the OMOP, it's got tables describing all of your patients, but on a particular level. So you have like a disease concept, you have disease treatments, so medical treatments or medical actions, certain amounts of phenotypic information. The phenopacket can be transformed out of these things. If you see the links I've given on the bottom, or these are software that has been written as part of the GA4GH to actually extract data out of these systems and to create the phenopackets from them. So the phenopacket, though, is it's focusing on outside of the EHR or messaging maybe from the EHR to allied systems, and for having very lightweight, for a simple way of actually exchanging data. So it's a simple JSON-based format you can fulfill this information with. So where does it fit in a kind of genomic medicine space? So the phenopacket is this bit in yellow that you can see here, and it's really a way that you use to describe a patient that you can store, take out of your EHR, and then move from into sort of a diagnostics case in this case. So what does the phenopacket actually look like? So what's in the phenopacket? So you've heard about what it is, but like, what really is it? So you see the line in black is the phenopacket concept. So the two parts above this is a family and a cohort. These are aggregates of other phenopackets. So you use a family would have probands, then you'd have mother and father maybe for the so you have a trio. You have pedigree related to this. So you have again sort of a packet of stuff that you can hand to somebody and go, this is my patient in the context of their family, with the genotypic information and the phenotypic information included. The cohort is much less well defined. It's basically just a collection that you can put any label on you to say that these phenopackets are similar in some way. So then we go back down to the phenopacket itself. So this is the individual level. So you're taking just a single patient. You want to be able to describe the actual individual, the diseases they might have, any interpretations that have been made upon this about this patient. So this would be the genomic interpretation. So this is one of the key parts of the phenopacket is the genomic interpretation side of things. From that, you've got measurements you can take from lab measurements, biosample data that you may have taken from biopsies, for example, the phenotypic features, which Peter's been talking about, which would be really where the HPO part sits in here, and then also medical actions. So again, this is the maxo part. So we're going to just have a quick look at focus on really these key parts. So these are if you take from the phenopacket itself, there's a kind of slots that you have within the actual schema for the message that you want to be sending to other people. So I'm just going to go through these in order and try and not bore you to death with the details. So we've got the actual patient themselves, the individual. So this is who we're talking about. With some fairly sort of innocuous identifiers here, when they're born, the phenotypic sex that they would ascribe at birth, and then also the vital status can be important. In this case, the case we're talking about is actually deceased, when they died, and why they died, and maybe for cancer cases, how long they managed to survive once they were diagnosed. An important part of the phenopacket is that because it's meant for sharing, not necessarily between health providers, but also externally as well. The requirement for what sort of data that gets put in there is relatively lenient. So we want to keep it so that you have as minimal information as possible as required to be able to share the data with other people. Sometimes this means you can be as explicit as you need to be, which would be in the case of a hospital where you're actually trying to have a diagnosis and for treatment. But in other cases, you might not need this. So I'll go into a little bit about that as we go on. But really, patient privacy and protection of their identity is a thing that we try to have in there. So the phenotypic features. So this is the bit that really leads on from what Peter's talking about. We're describing the patient's gestalt phenotype here, so that all of the phenotypic features that the patient has. In order to do this, we're using the HPO, and so this is a recommendation that we use the HPO here. You don't have to use any particular ontology or any terminology within the phenopacket themselves. So it's completely agnostic as to what type of data you put in there. But we have recommendations for the sorts of ontologies and terminologies you should be using. So the HPO is recommended for the phenotypic features because it's really what it's been designed for. But when you say to somebody, these are the phenotypic features of the patient, there's a lot of color that you can add to this. You can't just sort of say X, Y, and Z. You might want to say a little bit more about these bits that you're trying to describe them. So, for example, here we've got clinodactyly. So you've got which is a strange outside finger, not the fifth finger. But you want to say maybe when or where this fifth finger is to the left or right hand. So we have a modifier that you can add to this to show that, again, using HPO terms. So you've got more and more information you can use to describe your patient as specifically as possible. You have not only the modifiers of like which side or laterality there may be in there. You have the actual whether or not it's an excluded case, whether you look to see that the phenotype is there and actually found that there was no phenotype. So you're expecting an abnormality, but you found that everything is fine. So you can say this as well. Obviously, phenotypic features come in different severities. So there's a sub-ontology for this within the HPO, and we have slots for this within the phenopacket as well. You also want to say when phenotypes were onset. So that, phenotypic features tend to, as patients' disease progress through rare diseases, they tend to have more severe phenotypes, and then there's phenotypes that get more as they get along as well. So you can say when a particular phenotype actually happens. And then if you're very lucky, you can have a resolution of the phenotype. So well, actually, after this point, there's no more abnormality. I'm just going to point out the age part in here. So going back to the how wanted to try and protect a patient's identity or for sort of stratification, you can have different time periods in here. So within the age range, the age slots that we have here is actually a concept of a time element. So the time elements can be very, very specific, such as an actual timestamp as to when something was seen. But in this case, a phenotypic feature, you don't tend to say, well, it happened exactly 12 o'clock on Monday. You sort of say, well, it's in this case, we've got an ISO 8601 duration, so back when they were 4 months old. So this is what P4M in this case means. You can also give them a range of so you can say between 6 and 12 months, or you can give it an ontology term to say whatever that might be describing. So there's a lot of different ways that you can use this. You can use the schema to say things in more precise or less precise terms as you need it. So moving on from the phenotypic features, you often say measurements of your patients. As I said before, the measurements tend to be laboratory tests. And here we've got a LOINC code being used. So if you see in so you see at the top of the assay here, we've got the type of assay that's actually used. So this is again, we're using identifiers about all of these things. So every single term that's being used is either an ontology term or some sort of terminology that's being linked to this. So here we've got a LOINC code, which is for left intraocular pressure. And the measurement is really quantitative value that you're trying to convey to users. So here we're saying that it's again using a different ontology term for the unit ontology for saying we have a level of millimeters of mercury and 25 millimeters of mercury. Because it's a lab test, we tend to know what the reference range for these things are. So here we've got the high and low range. You can see that this is a little bit high. So we have so we said there's a kind of a link between, say, the measurements and the phenotypic features. So there's things like the LOINC to HPO projects that Peter's been working on, to where you can take a long term and then it'll produce a set of HPO terms maybe from that. So you have elevated light pressure. To that. And there's another measurement out lost that. So this can be really any measurement you might need, including kind of random things like red or white, yes, no, whatever's required in that particular measurement. The biosample is a kind of a little mini phenopacket in itself, but it's not to be used for the entire individual, but actually for biopsies that might have been taken from the patient. It's probably more of a sort of a cancer focus. As you can see, well, if you've got it's very small something. Down here we've got some histological findings or pathological findings that you can notice. So there's a tumor progression and TNM findings that they need from that particular physiological sample. But also with this thing, you need to know, really what is it that it's been taken. So here we're using the ontology, so this is to describe the anatomy. You've got phenotypic features of that actual biosample itself, and then measurements that have been taken from this. So in this case, we are looking at the. Oh, I can't even read it myself. Sorry. Oh, okay. This is the size of the tumor sample that's taken. And then notice down here that we've got the procedure, so how the actual sample was taken as well. So we've got a code here for the FCIT that we're using for to say that it was taken from enucleation of the eye, and then the site that it was taken from, so it's the left eye, and also where it was performed. So there's hopefully enough provenance in here that can be useful for the diagnosis, or just in the regular hands. And then the last thing here, we've got files. So there was a tumor sample was taken, and there's a sort of actually linked to a PCR file that was for this particular biosample. Probably the most useful thing for you guys, I think, would be the interpretation message. So this is once you've in part of the lab that we talked about. So we had the test would be sent off. You'd have the VCF sample would be all of the sample we taken, turn into VCF, for example, and then you'd have the phenotype packet itself used to describe the patient. So if you take these two together, you can then say, well, okay, so knowing this is the patient's genotype and this is the patient's phenotype, how do we then maybe come up with an interpretation to say this is, I think, our causative variant? So this is where the interpretation comes in. You can say here that we have a progress of the interpretation, so you can have something to say that, actually we're still working on this, but in this case, we have a solved progress. And then there's a diagnosis that you've created from this. So here we have a diagnosis of retinoblastoma. So we're using an NCFT term here. Probably mono would be preferable in this case, but we start with the NCFT for quite lots of time. And then moving down from there, we need to know really why the diagnosis was made. So we have a genomic interpretation. So this is a really unique part of the phenotyping. Here we actually say, we think we're pretty certain that this is the case, and this is the causative variant of this particular disease. And then there's an interpretation of the variant itself. And you can see here that let's start the analysis. And this is a pathogenic interpretation. It's just using the HCG classification. And then there's also some, you can say, well, now we know this, can we do anything about it? So there's a therapeutic actionability, which in this case, somewhat can be. And here we really it's got the variation descriptor. So this is the description of the variant itself. And this part here is actually using the variant presentation schema from the GA4GH. So this is another GA4GH standard that the phenotyping uses. And here we're showing there's a complement variation, and basically a knockout of a large chunk of life allele. So that gives you the idea about how the interpretation might be used. So this can be embedded in the phenotype itself to say this is what happened to the patient, and this is what we think, why we think they what we have diagnosed them with based on their phenotype. So moving on from there, once you have a diagnosis, perhaps you can actually do something about it. So we've got some medical actions that you can say. So we have as the patient goes through the system, they've got their diagnosis. What can you do about these things? So the medical action is there to describe as really any kind of procedure that you might want to make on a patient. So in this case, we've got some therapeutic actions and some actual treatments with small molecule treatments, and then finally an actual surgical procedure. We also have radiology, radiotherapy treatments that you can have as well. So they can all be described. They're all slightly different ways of recording information depending on what's required to describe that particular treatment. But in this case, so we're just going to go through the treatment of the agents. So it was given a particular drug.
Well, question.
Yeah,
I can see how this pulls together and reflects a patient that's been treated by a particular organisation, but it's probably especially relevant to GOSH, where patients have been treated maybe in a local hospital, have been diagnosed and referred out to Great Ormond Street, and then maybe back again for full-on treatment and kind of management beyond their care at GOSH.
Mm-hmm.
How do you deal with a scenario where you've got a phenotype that's actually reflected across multiple organisations? Is the way to put it together, or is it kind of a snapshot in time of a patient's care within a particular institution?
Yeah, I mean, there's no kind of mechanism for being able to do this. So you do have to literally just say, well, this is what this organisation has got from what they've recorded about the patient in this case, and this is the other one, and then you have to pull them together and then try and make the match.
Yeah.
But hopefully, if each organisation has actually started with the same phenotypic packet, they can fill in their own bits. So you just have diff basically, where you can say, well, this is what they did and this is what we've done. You've got the timestamps in there as well, so you have temporality included. So you can, as they go along, they shouldn't clash because you've got an actual timestamp of when something has occurred. And then because it's a file-based thing, it's very easy just to sort of to do a diff to see what the differences are. So as long as you start with the same thing and keep them in a kind of, you don't alter bits that you've already been given, then you can very easily put them together and make a sort of consistent history that you want. Yeah.
And how do you deal with incompleteness in a record? Because obviously you kind of described in one of your opening slides around the complexity of where the data is coming from.
Mm-hmm.
I mean, I work for a company that works with like gosh, pull together regulatory grade research data sets for analysis and so on. And one of our key challenges is understanding where data is incomplete. So it's potentially available, but it's not within the record. And I guess for this, is there a way of identifying or communicating to whoever's receiving this that there is missing data and it may not be complete? Or because I guess is there an expectation that this is an accurate record of exactly what's going on, or is there going to be always an expectation there is going to be gaps in it depending on where data's being fed into?
Yeah, that's a good question. We hadn't really thought about how to say we don't know.
Yeah.
That is a quite interesting thing to say. I mean, I'm guessing these things you can take them again instead of having to have an entire record as a phenopacket. You can also just use the individual elements themselves as standalone bits of information. You'd have to tie them together with an identifier to say, well, this is the patient. Well, actually, you'd have to put them within a phenopacket, but it would be a very limited phenopacket. So, for example, you would just have an identifier for the phenopacket, the individual identifier, as long as there's some way of matching those together between organisations, which I guess is the hard part. You can then just say this is the medical actions we took. If they had a procedure happened in one hospital, but then they wanted to, send that back again to another hospital to say, add this to your record, please. So, but otherwise, no, we don't really have any ways of sort of saying we don't. This bit we have it, but we haven't included it. So that's a part. Yeah.
Thank you.
Okay. I'll probably move on from here. Okay. So then, getting close. So we got again. So you've got all this information about what's happened and when it happened, and who did what. Maybe not who did what, but you have this information about all the treatments and procedures that have happened. Oftentimes, you might want to record what information you have into a computer system, such as again we have genomic analysis here. You have a VCF file or some other files you might need for doing this. And so you can just link this within the phenopacket itself. You have the URI here, so this can be either on a local system or in a bucket somewhere, like on a web service. So you can really look up this thing, find it, and actually use it. There's the file attributes here is a free text sort of map of key values. It's really up to you how you want to use them, but we do have a recommendation for when you're using VCF files and genomic analysis that you actually put genome assembly and what the actual file format this is, because it's going to be quite important. You can't always tell what the genome assembly is based on what the information you have in the VCF file. You have the wrong assembly, this is very wrong data. And lastly, the metadata, which is really just to talk about the terminologies and ontologies used within the phenopacket itself. So I've showed you quite a lot of different terminologies throughout the talk so far. This is really just a way of cataloguing all of those so that you can look up each of the identifiers used. So actually, this one's kind of weird because we don't have the HPO, which is very nice. So, for example, the NCIT, which is it's called the NCIT thesaurus. This is a URL that you can actually download that particular AL file that you use for the ontology. This is the version that was used. And this namespace prefix here, the IRI prefix, are two specialized fields. So when you see this NCIT prefix or NCIT colon bunch of numbers, you can then replace this prefix with this bit, and you'll be able to go straight to the actual definition of what that is. If you have the alpha file itself, you don't have the entire ontology, and this is where you can then start to utilize algorithms like Peter's just been talking about, so have fuzzy matching, where you don't necessarily have to have precisely the same term to know that you have something similar. So this is a lot of things. I don't expect to go through these. I'll just tell you what they're about because it's going to start at the top. This is the most important bit if you're interested in phenopackets. This is the actual reader docs. So this is the documentation about how to record things and what should be used for what I've just showed you. If you're feeling flush, you can get the ISO less version, which is like the 4454. This will cost you about 200 Swiss francs. So you could just get the free one. Also works, but if you like ISO, then this is the place to go. I'm being made so like this. This is something, one of the first things that happened between the GA4GH and ISO, where we, instead of making two different standards, we actually collaborated together to create a single standard that everybody's sort of happy with. The GitHub issue, the GitHub repository for the phenopacket schema that's here. This is really the reference implementation, and the schema itself is actually defined in a format called protobuf. It's very easy to define schemas like this, and that should be really, if you're going to be using anything, you should generate all your data from all your code from the protobuf definition itself. But we have reference implementations already. So for example, on there's the phenopacket tools and the Python tools that Peter's already talked about very briefly. These are Java and Python tools for using them either as command line or as libraries themselves, so that you can incorporate them into your own code. Really for creating, converting, and validating phenopackets. So if you want to do validation, basically you want to make sure that you have got terms that actually make sense, terms that are legal, that you've got all the data is actually matching up. So if you have a term in there, have you got the metadata to describe how that term can be found again? So this is kind of maybe not very exciting, but very important stuff to have. The more recently, then we've got the phenopacket store, which is what Peter has been working very, very hard on. So this is an amazing collection of phenopackets of over 4,000 individuals with Mendelian disease that he's been curating from the literature. This is a great resource. So hopefully there's more individuals added to the study. The power increases, and we use this for benchmarking as well. And then the phenopacket lab is something I'd highly recommend you look at. If you are don't want to do or can't use any kind of programmatic way of building a phenopacket, you want to describe somebody, a patient, any way, shape, or form, then if you go to the phenopacket lab, at phenopacket.dev.jax.org, you'll have a very nice user interface where you'll be able to create your own phenopackets. You can put in the terms that you might need. It has some text mining features as well, so you can just paste in a bunch of or write your own clinical report, and then we'll try and pull out the relevant HPO terms that you might want from that, and then add those into the phenotypic features. And then you can actually download the phenopacket afterwards and use that as needed. It's just a quick thank you for a lot of the people who are involved in this. There's really hundreds of people have been involved in this over the last 5 or 6 years. I think it's been going on. These are some of our users down the bottom who are the sort of the larger projects that have actually said that they're using it. It's quite hard to know that people are using it because unless you've received a phenopacket from them, you don't necessarily know what they've told you they're using it. So, but yeah, the European Genome Phenome Archive is a relatively newcomer to this thing, but it's great that they're actually using the phenopacket as a schema. But beforehand, they had no schema at all to accept data from users, so the data could be quite a mess. But hopefully, the phenopacket can help to sort of make that data a little bit more usable. All right, so I'm going to switch gears a little bit, and we're going to go into talk about Xmizer. So we've talked about how the HPO, Peter's done an amazing introduction of the HPO of really how it's organized and how it can be used in a kind of very technical and focused way. But that's just the phenotypic feature part of things. Really, when you're going to try and do rare disease analysis, you want to have some genotype information to actually back up your thoughts and ideas on what the phenotype may have come, where the phenotype may have come from. Again, this isn't all my own work. There's a lot of people who've been doing this over the years. Just to sort of put this in context of where we're talking about as to where where Xmizer fits in sort of the general workflow of the potential diagnostic sort of pipeline, that would be the blue part here. So what is the Xmizer? It is a Java-based command line application, and it's really just a very simple job of annotating, filtering, and prioritizing variants for rare disease diagnosis. There are multiple genome assemblies that you can pull your variants against, and we support both GRCH37 and GRCH38. So really anything that Nomad will support is the gold standard where we're trying to do. We're looking at, we can analyze either whole exome or whole genome sequences using long read or short read data, and we are also able to co-analyze structural variants and small variants at the same time. So if you've got cases where you've got compound heterozygous alleles, we can detect these. I think we've had a couple of cases in the 100,000 genomes project, so where this has been the case. So the annotations, it will actually use things like the variant effect predictor. It doesn't use that itself. It uses a Java version that we have actually internal to Smizer, called Janavar, which is came out of Peter's group. So that's sort of really the starting point, is to say, what is my variant like? It then will do variant frequency annotation. So it uses Nomad and the UK10K, the ESPs, the exome server project, and also TopMed data that comes out of the DB state. For variant pathogenicity, the scores we use are Revel, MVP, Alpha Missense, Splice AI for the kind of the really new versions, the sort of second and third generation, and then also the kind of the earlier machine learning models such as CIF, Polyphone, Mutation Taster, REM, and CAD. It's a bit unfair about CAD. CAD sort of keeps up to date with things, but and then for the patient phenotype matching, we use the HPO. And this is probably where all the magic happens. So if you take one thing or another out of context of each thing, you end up with not a very good picture about what you might have. So for example, if you have a patient phenotype and you just use something like Phenomizer, you'll get a lot of information about potential diseases, but you won't know for sure because you don't have the genetic information to back up your diagnosis. Likewise, if you have just the genetic information, you're filtering on this thing, you'll find that you have thousands and thousands of variants left over, and you have no clue how to stratify these things because they can all look quite equally pathogenic. So really, the HPO is a very, very, very key part of this, and the most important part is to be able to put the two together, so you have an idea about what you're looking at. So I'll just sort of repeat how this is working here. So this is the main baseline workflow that we have. We're going through the VCF side of things first. So this is the variant side here. So we're taking a VCF file. We can either have a single sample or multi-sample VCF if you have a family. And then this is basically goes through with annotation, like I said beforehand. So we'll be saying, well, what is the pack? What is the variant? It's the variant as well. So is it on target? It's off target. So is it in an intergenic region? Maybe this is not so much to start looking at, but in the first place, so we can say look at the coding regions to start with. We'll then maybe remove all the common variants. We look anything over 2% would be less informative to look at. And then we start to classify the variants themselves based on the predicted pathogenicities, such as Revel, Alpha Missense, or Splice AI. That's this part here. And then also the mode of inheritance is incredibly important coming into it. If you have an allele which is just not in the wrong allelic state, or a variant in the wrong allelic state that doesn't match the expectation of what the disease ought to be, then it's hard to say that this is the right variance. So we have, also we have sort of quality filters for the VCF itself. So if you want to have particular quality thresholds that's in the VCF file, or whether or not the variant is actually passed the VCF filters, then you have those to start with. These are quite important to run. Turns out we get rid of a lot of false positives. We can also use it to say, actually, I only want to look at this genomic region, or you can say, I only want to look at this particular variant, these particular genes. So you can just basically use it as a way of doing some gene panel. Wouldn't recommend doing that necessarily, but that's a possibility to do these things. We've also got the ability to say, well, this is a regulation feature, so anything which is in an intergenic region that you might want to look at will also be possible. So it's talking about gene symbols. You can just have blacklists or blocklists as well. So it's saying, well, actually, I don't ever want to see anything from this gene because we just know this isn't a gene involved in rare disease. And then finally, this is phenotype profile match. So if you look at the green side here, so this is where we're taking the HPO, and I'm going to do a massively simplified version of what Peter talked about already. So we're taking the patient phenotypes and then we're matching them basically against reference diseases from the HPO annotations. And then we also take information from the MGI and the IPC. So these are the mass phenotype based bases. So if you don't have any information about a human disease gene, you may find that there's a new disease gene association that we haven't actually found before, that you'll get that information from a mouse knockout. So here we can actually do not only just human disease, but also human to mouse or human to fish or protein-protein interaction networks. So once you've got those two bits of information, you can combine them together to have a final prioritization score, and then you have a rank list of variants that come out at the end. You just start at the top and then move down. So Peter's already told you more about this than I'm qualified to tell you out. Of course.
Why compare to fish?
It's just another source of information. We have found that, I have to say that it hasn't been the most useful source of information, but it's something that we've had for right since the beginning of maximizing, which is really just, if you have, so different animal models can slightly be used for maybe different types of phenotypes. So I think eye phenotypes are quite prevalent in fish, so they could be more informative for those particular phenotypes if you don't have a human phenotype match already. That's it.
We better, I'm just thinking in terms of time, maybe to be question.
Yeah, we can maybe have this.
Yes.
Yeah,
Yeah.
Yeah, so as Peter was saying, we've got the HPO, it's a tree. You tend to walk down there. If you got to the bottom of the tree, these are the more specific terms in terms of what the actual phenotype is. As you go up, you're getting less specific. So no matter where you put your phenotype terms, phenotypic features for the patient description, you want to make sure you're down bottom here, and then Xmizer can match anything that is higher or ranked up from here. So as Peter already told you, much better way. The real way that this works is information content. So as you can see here, we've got the trees being coloured in this case, so that the lighter the colour, the lower the information content. And as you go down here, you can see the colour gets darker and the information content is getting higher. And this is here because we've got the inverse log of the frequency, so in that particular corpus. So if you see something with telecampus or hypertelorism, then you've got a very high information content. So if you can annotate something down here, you're going to get a much more specific hit than if you had something up here. And there's no point in annotating this and this because this already includes the markers. Peter's already shown the fuzzy phenotype matching example, slightly different colours, but it's the same idea. So as I said before, the phenotypes can present quite differently, but you may have the same actual condition. So the idea with Xmizer really is rather than having a kind of a panel approach going, well, I think it's definitely this, it's one of these things. All you have to do is you just have to say, these are the phenotypic features of my patients. There's no kind of bias included in this thing because we take all the information that you might need to know about as part from the HPOA itself. And people you just have to say, it's like Dusty Bin, right? You just say what you see, and then the Xmizer will match your patient phenotype to the known phenotypic features and rank them accordingly. So these actually might have the same phenotype score, sorry, phenotype similarity score to the body-matched donor syndrome, but they have quite different phenotypes themselves. So we like to make sure that we have actually, trying to be as unbiased as possible, and to make sure that this is really working as we hoped for. And we've had some sort of an independent group in the Children's Hospital in Los Angeles has actually worked Xmizer into their diagnostic pipeline. So that was a lovely thing to see when we were just looking through the literature to see that they had included this in here. And they actually found that they had a lot of cases that they had been ranked as one of the top hits were not actual known disease gene associations, but Xmizer managed to find them due to mouse hits, and then they were later found to be causative through different literature and different diagnoses. We've been through this in some benchmarks. Sorry, I won't go too long here. We've been used as part of the genomic medicine service since it started. So Damien's been working with GEL, and it's part of the 100,000 genomes pilot. We found that it was if you see here, so this is the disease panels, but this is used for the bacterial process themselves. We had a kind of an automated panel assigner. Features found that maximizer basically performs in this is the top hit for maximizer. So if you took the top hit for maximizer, you get 75% of your diagnosis. And if you make the top 3 or the top 5, you basically you're going to be getting as good as you can possibly get. The downside of this is you're going to have a lot of variants to look at potentially if you need to go down this study further. So if you put the panels together, you get a much better predictive value of these things, but essentially, excellent panels work quite well together. We've also been a top performer at the KG. So the KG is the Critical Assessment of Genome Interpretation. So it's a very Genomes project that was run by the Broad. And Xmizer was one of the top performing algorithms in this one. But this is our test set that we were given when we knew what the actual result was. We found that we were able to find basically almost all the cases in the top 10. So 28 out of 35 were top set for if you use it in a disease, like four model, which is using mouse models, like human only model, which is slightly better for the sort of diagnostic purposes. We've got 31 out of 35 cases at the top here. We're addressing this at the moment with some new phenotypes and measures. And then when we go to the actual diagnosis, we've got out of case out of 14. We've got 6 out of 14 in the top there. So these are completely undiagnosed cases that were missed and sorry, they weren't missed, were not released in any way, shape, or form to the environment. So this is a blind test. Just to go through very briefly about Xmizer and how it's developed and what we're doing. So Xmizer has been around for, I think it's been since 2012 when I first released it, been around for quite a long time now. And then we have data releases and we have software releases. So the data releases are named after the year, so it's 2019, and then month or 2 for February. So we tend to have 2 releases each year for data. And the data is mostly compatible once you go along with each different release of the software, but not in this case for version 14. So we use semantic versioning for the software itself. So if you go from 12 to 12.1, you should be pretty safe and the things won't be changing too much. If you go from 12 to 13, you can expect some major features, probably some breaking changes, depending on how you use it. The command line itself has not really changed at all over the year, since 13, so since 2021. It's not to say that it won't, but it had tried to keep it as stable as possible because it's kind of pain if you have to recheck things. We mostly have a release every 6 months or so, depending on what it is. This release stage, we're probably going to be changing it soon, so we have more frequent releases in line with the HPO annotations, because this is incredibly important to keep up to date with these things so that you actually know that you're going to get a diagnosis and not miss anything based on the latest data. When it comes to the genomic data itself, so the variant frequencies, they don't tend to get updated as often because we're talking about things like Nomad, which are huge projects, and they release maybe once every 2 years or once every year or 2 years or something like that. I won't go into too much detail here, but basically since 13.1, since 13.2, we have the combined structure and small interpretations. But since 13.1, we've already been focusing on adding the ACMG annotations. So the ACMG annotation, does everybody know what the ACMG annotations are? No. Okay, so ACMG is for the American College of Medical Genetics. So they have a set of guidelines for of how to assess variants, so the sequence variants in such patient group. And given they have different categories. So here you've got something like the PBS1 category or PP4. They're all kind of a little bit obscure in terms of the terminology used, but basically P for pathogenic. And then you've got VS for very severe, or S for severe, M for moderate, and P for supporting. So they all have different weights that you can give them. So the idea is really that you can work through a set of the ACMG criteria to then say with a certain amount of confidence or not that your variant is indeed pathogenic or benign based on the patient's phenotype. So I've been adding these. There's quite a lot of different categories that need different formats, different sorts of data to be able to assign them, but I've added as much as I can as I'm going along, and we keep adding more. So we've got pretty good coverage. There's another there are different working groups within the ACMG for particular types of ACMG classifications. So there's the original version came out in 2016. I think that's the Riggs et al. Paper. But then there's subsequently been updates to this to say that actually you shouldn't be using this. You should be using this particular sort of weighting algorithm. So we've upgraded this to use the Bayesian point system that they came up with in version 13.3. And then in version 14.1, which is in development at the moment, we're going to be using the splicing recommendations. So for any splice variants that you might find, these will be more easily automatically classified by Xminder. Just to show how this sort of works in action, which is when Damien was wondering what to do with himself during the pandemic. He decided to look at the undiagnosed cases, for the unsolved cases from this is from the pilot project, obviously the actual that whole 100,000 genes. Okay, this is from the whole 100,000 genomes project, so the 25,000 or 24,000 unsolved cases. And we just thought, well, what if we just take the easiest possible things, the most likely things to look at? So we'll also look novo variants, loss of function, clinical pathogenic variants, take those to start with. Then we take the phenotypic similarity between the proband and the disease gene, or a green gene on the latest disease panel, and then filter those again. Then lastly, we take the Xminder automated ACMG classification. So you can see there's quite a big difference in terms of from about 2,500 to 980 variants. Then we have some additional manual review to remove variance constants. Sorry, I'll do this. Additional manual review, and then Genomics England's clinical laboratory actually went through, the genetics lab actually went through these variants and then came up with a diagnosis of these things as well. So we had an additional 467 novel diagnoses based on this. As you can see here, this is my last, probably the most important thing I'm going to say here. As you go along, these are different Xminder releases. So these were the undiagnosed candidates. As you can see here, there were no new diagnosis. If you reran Xminder using the new version of Xminder, the latest version at the time, with the data, as you move the data through, you can see you're getting more diagnosis occurring. So this is a reinterpretation here. So basically, the takeaway message of this thing is rediagnose the variants as you go through, and in cases for any undiagnosed cases, as new disease gene associations come out, the HPO will add these in there. We have new phenotypic features associated with those diseases. You can then actually start to diagnose cases that you would have been able to would not have been able to find previously. And in cases here, you can see we've got like the precision and recall of these things. If you add the ACMG categorization on top of this, you end up having much better precision. So you're getting up sort of 87% with only very small loss of recall. So just as a kind of a very quick thing, again, like I said, don't miss the settings. It tends to be quite good to start with. If you want to be as specific as possible with your phenotypic features for your phenotype terms that you're using, go for the lowest or highest IC that you can find, the most specific terms that you think is going to be useful. Don't overlook things either. So you can often find that maybe not enough features are being put in there, and you think, oh, that's not important. Can be the case, but a bit of an art form to this. As Peter said, there's a publication how to do phenotyping correctly. Keep the software up to date. It's a very important thing to do this, and also keep the data up to date. You can find new diagnoses that you would have missed beforehand if you do this. Okay, thank you.
Thanks very much, Jules. That's covered a lot of ground there, and again, I'm sure we've got loads of questions, but we better hold just because I want to make sure people have got time for a cup of tea or coffee. So if we take a short break now, shall we plan to come back? It's just, we make it 10 minutes, so we can make it 20 past to start the next talk. And as I say, we'll hopefully have opportunity for questions later in the day. Thanks very much.